Good morning, everyone. It's great to see you all here today. I'd like to echo Mark and Alice this morning for welcoming you all to this conversation. And as our panelists on this first session noted, there are just an amazing amount of unique opportunities that are actually in practice today that can help improve biomedical innovation. What we'd like to do now is turn the conversation to the underpinning data around all of this and ask important questions like how can we harness data throughout the discovery, clinical development, and regulatory review to better understand the trends and driving factors towards innovation. Is there a better way to approach these questions? Are we taking a fully informed approach to understanding what the true drivers are to innovation? And also, how can we move beyond simple counts of new approvals each year to really understand if these policies are having an impact on developing better innovation? And then finally, how can we actually track in the post-market environment to really understand true value and understand if these new innovations and policies intended to incentivize development of new innovations actually result in better outcomes for patients? So I'd like to introduce our panelists for today. We're going to do it a little bit differently than we did on the first panel because our panelists on this session have a lot of great analyses and results to show you all. So in order to see the slides, we'll have each of the presenters come up and then go ahead and sit back down off the stage until all of the presentations, and then we'll invite you up for the panel discussion. But first with us will be Mr. Jonathan Leff, who's a partner at Deerfield Management and chairman of the Deerfield Institute. After Jonathan, Dr. Murray Aiken, who's a senior vice president at IMS Health and executive director of the IMS Institute for Healthcare Informatics. Next would be Dr. Peter Newman, who's the director at the Center for the Evaluation, Value, and Risk in Health at the Institute of Clinical Research and Health Policy Studies at Tufts University. And then finally, Dr. Marta Wazinska, director in the Economic Staff, Office of Strategic Programs at the Center for Drug Evaluation and Research at FDA. Welcome all of the panelists, but we'll start with Dr. Jonathan Leff.
Thank you, Greg, and I want to thank Brookings for hosting this important conference once again on the state of biomedical innovation. You know, I think this is my fourth year being invited to speak here at this event, and I was thinking that each time, remembering that each time for the last 3 years, Mark has suggested that, hey, it would be great if we could discuss not only what is the state of biomedical innovation and what should we do about it as a policy matter, but how do we measure it and how can we measure it better? And then I realized that in each of the last 3 years, I've pretty much politely ignored that request and really just spoken about the state of innovation and what I think could be done about it. And the reason for that is that I couldn't quite figure out what to say about the question of measurement other than that existing measures are not very good, but they're what we have to work with. So the good news for me is that Mark and Greg have invited me back this year despite my persistent refusal to talk about the topic at hand. And the good news for Mark and Greg is, and for all of you, I think, is that I am not here today to talk again about my opinions about what to do about biomedical innovation, but I actually am going to talk about the question of measurement. And the reason that I have something to say about the question of measurement is the direct result of a tremendously productive collaboration that our group at the Deerfield Institute has had over the past year or so with our friends and colleagues here at Brookings on this specific question, where we've set about not only to think about what are the issues with measuring and tracking biomedical innovation, but to actually do something about it. And so that's the topic that I want to get into today, is what are we actually attempting to do about it. So the topic I want to introduce today is the project that we're undertaking, as I said, at Deerfield Institute in collaboration with Brookings, and it is an effort to build a more comprehensive database tracking new drug innovation than has existed in the past. Just in the way of background, I think, as everyone here knows, there are metrics that are tracked and commonly cited to assess the health of the new drug innovation enterprise. And in fact, my talks here over the last several years, I think, have started with charts like this, that look at, as I think Greg just said, one of the most commonly tracked metrics, which is the number of NME approvals, and it's often plotted as it is on these two slides in conjunction with industry investment in R&D. And these two metrics, if you looked at them over a couple of decades on the right or over the period of 6 decades here on the left, in all cases seems to point to a decline in R&D productivity. Basically, we're spending more and more, and it's not clear that we're getting more for it if you just look at the output of new medicines being approved each year. So these are the kind of metrics we have, and this is something that people spend a lot of time talking about and looking at, and it does paint a concerning picture of the state of biopharma innovation. And I think that perhaps the greatest value of these metrics is these metrics have helped to get the attention of policymakers. And policymakers, as I think people here know and was just discussed in the last panel, are looking for solutions because of the apparent decline in productivity that we're talking about here. I think people here are familiar with the FDA Safety and Innovation Act, or FDASIA, in 2012, where a number of reforms were implemented designed to enhance the innovation process and accelerate drugs to market. Also in 2012, the President's Council of Advisors on Science and Technology undertook to dig into this and made a number of policy recommendations. And as has been said, Congress is now looking at this question in the 21st Century Cures Initiative through the House Energy and Commerce Committee, and the Senate just this week held hearings on the topic of how to advance biomedical innovation. So there's a focus on the topic. And on the one hand, it's a great thing that these kinds of broad measures of biopharma innovation have contributed to a sense of urgency among policymakers. But on the other hand, the dialogue about how to enhance new drug innovation highlights the limitations of the existing metrics we have. So while we can say that R&D productivity appears to have declined over the course of 6 decades, we don't have great data to allow us to answer the kinds of critical questions that inevitably need to come next. So for example, what has caused the decline? Lots of different views and different opinions on that, but actually not great data to get underneath the question. Is the decline more acute for certain classes of drugs or certain therapeutic areas than others? And what has been the impact of prior policy interventions that have been intended to help address this problem? And then probably most importantly, and what is the ultimate question, is as we talk about new policy interventions, new ideas that sound good, how do we think about and measure what their effect is likely to be, and then actually track the effect that they have if we implement them? So in effect, because of the state of existing metrics, we're left with this problem, which is how can we fix it if we can't measure it? So when our groups at Brookings and Deerfield began to dig into this problem more deeply, we first studied the literature to identify the most commonly cited metrics that are used to track the innovation process. And there are many, such as NME approvals, R&D spending, venture capital investment, or other forms of investment, new company formation, FDA performance metrics, people look at review times or first cycle approvals, and then measures attempting to track the cost of drug development or the success rates in drug development. These are the major categories of measures that we found out there that are tracked or attempted to be tracked and talked about. While all these types of metrics have made valuable contributions to the field, we identified some key limitations with them. In general, these metrics paint an incomplete picture. For example, like the aggregate measures of biopharma productivity that I showed you at the outset, these metrics in many cases may provide an overall picture of what's going on in the enterprise, but they don't allow for disaggregation to analyze and understand the underlying drivers or what's happening in different components or different parts of the ecosystem. We also found that these data sources are often inconsistent or incomplete when you look at different data sources that may be trying to track the same thing or different aspects of the same thing. Some of these metrics, such as cost or success rates in development, are often drawn from surveys, and thus, while they provide meaningful information, they may not reflect the full landscape. So they may, for example, track what's happening in big pharma, but not what's happening in smaller biotech companies or vice versa. And then of particular importance, many of these metrics are not routinely collected and updated. What you find in the literature is that an individual researcher or a group of researchers assembles data from a bunch of different places to try to answer a specific question. And then they're able to say something interesting about that specific question. But the underlying data then that was created quite laboriously by those individual investigators is not frequently maintained and is not available for future analysis. And then even when it is, the underlying data is often not publicly accessible to future researchers who may want to build on published analyses or dig deeper. So we have quite a fragmented system, and one that requires actually an enormous amount of resource of individual researchers to answer questions that one would have wished could be answered much more easily, and as a result, there are a lot of questions that simply go unanswered. So our teams at Brookings and Deerfield set out to create a comprehensive database of key development, regulatory, utilization, and outcomes characteristics of each new drug that's been developed over the last 20 years. And we're doing this because we thought, wow, that should be out there, and if it were out there, a lot of these questions could be answered with empirical data and answered much more easily than they can be today. But it's not out there, so we said let's set out to do it with the goal of enabling not just ourselves, but the entire research community and people who are interested in the topic of innovation to be better able to measure it and better able to interrogate the data to think about what the impact of different kinds of policy interventions might be. So we published the rationale for this project and the basic architecture of our proposed solution, this database, in Health Affairs last month. And the primary objective of the project is to develop, populate, maintain, and make publicly available a comprehensive repository containing key metrics of new drug development, utilization, and impact. Now, we recognize this is a big and ambitious project, and we have lots of ideas for it, so of course we have to break it down into stages. This slide shows on the left our 4 proposed research categories and some of the initial data elements that we intend to populate in the database on the right. And I apologize for the eye chart here. I won't have time to go through this in detail today, but these exhibits are taken directly from the Health Affairs paper, so you can refer to them. The main points that I want to draw people's attention to here today is that first, we ultimately want this to be broader than just looking at the development characteristics of new drugs. That is where we'll start. We'll start by looking at key development characteristics such as development time and clinical trial size and regulatory aspects such as different regulatory pathways to be able to look at how all those things interact with the health of the innovation enterprise. That's where we'll start, and that's really perhaps the low-hanging fruit for drugs that have been approved by FDA over the last 20 years. That's a big project in and of itself, but we do intend to go beyond that. That's really research category number 1, phase 1. We intend to go beyond that and look also at measures of new drug utilization and outcomes, and also at drugs that fail or are abandoned in development, as well as those that are approved. And finally, at broader macro or system-wide kinds of factors that affect the innovation process. The second thing just to highlight here is that we know that what we're talking about here is a big effort. It's a big project and it's a vision. It's not something that's going to be completed overnight. It will be developed in stages, and hopefully those stages will be useful to the community to conduct analyses as we build it. But this is really a vision for something that we hope to create and build upon for many years to come. So how are we going to make this a reality? The first step is the commitment to do it. Brookings and Deerfield have developed the concept with input from a variety of stakeholders, and we're now committing substantial resources to it. Second step is collaboration. We are looking to build a consortium of stakeholders who share this vision. The third step is to access the right expertise. We need to build a consensus around exactly what metrics are important to collect and what's the best methodology for doing so. And fourth, we want to get this right. So we intend to make the necessary investments up front in data sourcing and development in order to ensure that the database is ultimately as accurate and as comprehensive as it can be. So finally, where do we go from here? I just want to close today with a call to action. We need everyone's help. We at Deerfield and Brookings can't do this alone, and we don't want to do this alone. We want collaboration across the entire community. To start with, we want to engage with key stakeholders who have an interest in this project and who can contribute thought leadership around how best to do it, how best to design it and architect it, and what the questions are that we should make sure we can answer when it's done. And stakeholders who can help us find and access the data sources to populate this and make it all come together. And then finally, or additionally, we're looking to establish expert groups to build consensus on research questions of interest and to help us design the database and to create definitions of data elements, because you often find that some of these things are not totally objective and require expertise from different fields within the community to be able to designate a given field for a given NME. So this is a call to action. We hope others will join us and we'll be able to build a database that will ultimately be publicly available for analyses to try and empirically get at the kinds of questions we're all talking about here about how to make biomedical innovation work better. Thank you.
Thanks. Good morning. I'm Murray Aiken from the IMS Institute for Healthcare Informatics, and I'd like to spend just a few minutes taking you through 3 examples of research that we've been involved in. At least a couple probably fit on that left-hand side of Jeff's chart, where there are limitations to what we're able to see, what we're able to report out on. And indeed, in the context of Jeff's comments, these are the sorts of areas where we look forward to being able to contribute to a broader effort to get our arms around this important issue of measuring the health of the biomedical innovation system. The first thing I want to talk about is a piece of research that was published in Health Affairs last year, last month. Lead author was Ernst Berndt from MIT. This, at some level, was trying to connect the issue of what's been going into R&D budgets and expenditure and what's been coming out in terms of economic returns for that investment. And at some level, this sets the bookends of the innovation ecosystem. And so what we sought to do was update some of the earlier work that's been done looking at the economic returns from investments in biomedical innovation by the CBO, by Grabowski and Vernon from the 1980s and 1990s and a little bit last decade. And what we were able to do was create a data set of 466 new molecular entities launched between the period 1991 to 2009, and look at what the commercial returns have been for that set of innovation. About 80% of the new molecular entities were small molecules, about 20% were large molecules, and what we were able to do was look at the lifetime sales for each of those molecules, where we defined lifetime as the period from launch until patent expiry, plus 24 months for small molecules, plus 60 months for large molecules. We did the analysis starting with the US market and then applied a multiplier for global markets. We converted everything to $2,005 and created a net present value so that we could see what have been the trends during that 20-year period or so in terms of the lifetime sales of the new molecular entities. And this is a summary of the findings. So this is the average net present value of the lifetime global net sales of the novel active substances, the NMEs, in 4 different cohorts. So we grouped around 4 to 5 year periods. And you can see from this that for small molecules, the average present value was 3.3 billion in the first period. It rose to 4.2, 4.8, and then a reasonably significant decline for the cohort of new products launched in the 2005 to 2009 period. For biologics, a stronger net present value, a higher net present value, and particularly high in the 95 to 99 period, which was a time when some very remarkable innovative biologics were launched and came to market. But then a fairly steep decline since that point, and in the most recent cohort, a lifetime sales of 2.7 billion. So the blue takes that combination, showing the average. And again, what is catching our attention, of course, has been what's happened in that most recent cohort period where the average net lifetime sales has declined. Now, again, this is a good example where we can measure what's happened. Getting underneath that and looking at the drivers of that change is more difficult, although we do have a very detailed database to work with. Things like orphan drugs, for example, which have become a more significant share of the new molecular entity launches, on average carry a significantly lower net present value of lifetime sales than do non-orphans, not surprisingly. So as that percentage of drugs coming through as orphans increases, not surprisingly, that's one of the reasons we see that downward trend. We also know there's been a lot of changes in the commercial market for new pharmaceuticals, heightened competition, growing consolidation of purchases, a growing scrutiny, indeed, by payers as to the value that they receive from new drugs. And we do think that those factors have been having an impact on the lifetime sales trends. What we also wanted to do then was link that lifetime sales to the costs associated with the development of innovative biopharmaceuticals in order to derive an economic return. We used an 11% discount rate for small molecules, 11.5% discount rate for large molecules. We used the Tufts average cost of bringing a new drug to market over this period. We used operating cost metrics from the literature. And so when we combine all of that, we end up with a net economic return, which, as you can see, was relatively strong in the earlier period, particularly for biologics, notably. But importantly, in the most recent right-hand cohort, 2005 to 2009 launches, we actually see, on average, a negative economic return, again, using an 11% discount rate for the average of the drugs launched in that most recent period. So this is one of the issues that I think a lot of people look at and say, what does this mean for the sustainability of investment into the biomedical innovation if there are, in fact, no economic returns? This is also the sort of analysis that, from our perspective, doesn't get looked at as closely or as frequently as it should, to understand whether, in fact, we have that right balance of incentives for capital to be put at risk into investment in biomedical innovation. A second piece of analysis we've done is to look at what's happening in the pipeline of preclinical products. So we maintain one of the, what Jeff would refer to as a less than perfect database, although I would say it's consistently imperfect, which means the longitudinal view can be relevant. where we seek to identify all of the products in both preclinical development as well as clinical development. And here, this is just a snapshot of what we see coming out of our database. This is looking at the number of preclinical projects where there has been activity within the prior 3 years, which is an important caveat. But one of the complexities of measuring things is knowing when something is really dead and when it's just dormant for 5 years, 10 years, or longer. But for the sake of this analysis, we simply looked at products that at the end of each of these 3 years was in preclinical development and had been active within the prior 3 years. And as you can see, it's a significant decline from about 5,800 at the end of 2004 to 4,700 at the end of 2009 to 2,000 at the end of 2014. Again, this is a good example of, well, these are the numbers, but what's really going on underneath that. And again, I think this is where there's still a gap in being able to pull this together and do a more causal analysis of what's really going on here. And whether indeed this is good news or bad news, because it's a little hard to tell simply from the numbers. This also speaks to the growing importance of oncologics in preclinical development. That's the orange bar in the middle there. That's while it's down in absolute terms, it's increased its share of the total activity, going from about 29% to 40% over the past decade. CNS, central nervous system projects are now 16% of the total, and anti-infectives are about 20% of that total. The other thing we looked at is who's bringing these preclinical compounds into the pipeline, and what's the role of big pharma in that. What we've seen is that the 20 largest corporations in biomedical innovation have reduced their share of the preclinical compounds over this 10-year period from about 15% of the total to 6% of the total. Again, this is one of those this is one of those facts that I think we should be able to talk about in terms of whether this is a good thing, a bad thing, and what the underlying drivers are. Just finally and very briefly, we've also been looking at the average time for clinical development to occur from stage to stage. And here we're just presenting the averages of products moving from phase 1 to phase 2, from phase 2 to phase 3, and from phase 3 to submission in 5-year cohorts. And then we've divided it between what we would call traditional therapies, mostly small molecule, more primary care, chronic kinds of therapies, and those that are specialty, more biologic, and so on. And again, the trend here is one that shows a lengthening of time for products to progress from stage to stage. So from phase 1 to phase 2, the average in the most recent 5-year cohort, 25 months, up from 18 months in the 2000 to 2004 cohort, 36 months for phase 2 to phase 3, 36 months phase 3 to submission. Again, these are the top line numbers. We have about 500, an N of 500 in the 2010 to 2004 cohort for each of the phase changes. So it's a reasonably good data set, and one that could be subject to a lot more analysis, again, to try to get under the surface here and say what's really going on, how much of this is related to policy changes, how much of it is related to the economic factors we've talked about, and the other changes that are occurring in the environment. So let me stop there, but again, we are very supportive of the idea of contributing and participating in efforts to bring together what really does look like a very disparate set of information that is around and about, which doesn't come together in a very easy way to date, so that we can have a much deeper understanding of the health of our biomedical innovation system. Thank you.
Good morning to all of you. I'm Peter Newman. I direct a research center at Tufts Medical Center in Boston. Thank you very much, Greg and Mark and Brookings, for inviting me today. People have been mentioning this $2.6 billion figure that comes out of Tufts. That's a different center than mine. It's colleagues, and they do great work. I direct a different center, and we've been in our own way trying to quantify value and innovation. So in my brief time, let me share with you some of the data we have and tell you how we're trying to go at this. So 3 basic research initiatives. 1, the Tufts Cost Effectiveness Analysis Registry. Some of you may know, a database of cost effectiveness studies. 2, a database of quality gains. These are quality adjusted life years, a way of measuring population health. I'll share with you some new findings. And finally, we've been tracking all of the Medicare national coverage decisions over the last 15 years or so. And we have a database of these. And it's another way at getting at how evidence is translating into coverage and reimbursement decisions and a window into the innovation process. So first, the Tufts Cost Effectiveness Analysis Registry. There's our website. It's a publicly available database. At least we make a good chunk of the data publicly available. It's a database of cost per quality studies. Okay, so we read through the vast medical literature, the peer-reviewed Medline literature. We collect data on every cost per quality study as it appears in this literature. We collect a lot of information. We standardize it in various ways, including in 2012 US dollars, and put it into a database. So this is the only math on the slides today. In the numerator of these ratios is at delta costs, the change in costs that result from investing in these technologies. I'll give you some examples in a moment. In the denominator is qualities, change in qualities. And the delta is because all of these interventions are being compared to something else. Status quo, next available, best available alternative. So you can, and let me just say, there are challenges with cost per qualities. We are well aware of them. We've written about them. The great strength is it gives you one number that captures morbidity and mortality in a single ratio reflective of value. With any single number, there are always questions about whether you're capturing well what people care about. Nonetheless, what you can do with these ratios is array all kinds of innovations and technologies and therapies, diagnostics, procedures on a graph. So on the left side, cost saving and health improving. And these are just examples. We have 11,000 of these in the database. Some are older and probably not as relevant, but you can search and find it. So vaccination against chickenpox of infants. One study found this was cost saving. Saves money because you offset enough cost to offset the cost of the intervention, the vaccine. And as you go up the scale, 20,000 per quality. So now it's, in a way, costing us money, costing society money. For every quality adjusted life year gained or produced, it's costing $20,000 in an aggregate term. Gene assay guiding chemotherapy in breast cancer patients. And as we go up the scale, it's becoming more expensive to buy units of health. In other words, less efficient ways of investing in innovation. Sovaldi, everyone's poster child for high-priced medicine, actually shows up, at least in some populations, as fairly reasonable value for money. Most people, and this is a debate and controversy we can come back to, most people would say $50,000 per quality is, under conventional standards, pretty good value for money. Now, that's not to say this is not a big budget item for the payers or not controversial in other ways, but if you simply look at the return on what you're getting in terms of a cost per quality, sofosbuvir in HCV, hepatitis C, is fairly cost effective. 65-year-old men screening for osteoporosis, less cost effective. Lung volume reduction surgery comes out as even less cost effective. You get the idea. Recently, Medicare covered CT screening in lung cancer. Shows up as fairly good. All of these ratios beg questions about which population and which comparator, and on and on. A lot of that detail is in the database. But you get the idea. It's a way of quantifying innovation. It's a powerful construct. Okay, let me turn briefly to quality gains. Out of the denominators of these ratios, you get a measure of population health effects, quality gains. And again, it gives you one number to quantify value and innovation. And so my colleagues and I have been doing this in various ways. We had a paper last fall in Health Affairs which looked at specialty drugs versus traditional drugs in terms of quality gains. And what you see, and you can read the paper for details, but a lot of the specialty drugs, despite high prices, which are real and in many ways eye-popping, the drugs actually do pretty well on the quality gains. In other words, they seem to produce pretty robust health benefits, at least compared to traditional drugs. And furthermore, if you actually then look at the additional costs and quality gains, the results are somewhat complex and nuanced, but a lot of the specialty drugs do pretty well. Not all. You have to read the paper. It does depend on the specifics. But again, it's a way of quantifying value and innovation. So this is brand new data, never before seen until right now, except by Mark McClellan. So we stratified the quality gains by FDA approvals over the last 15 years, since 1999, given FDA designations in different categories. So FDA designates certain drugs as fast track, accelerated approval, priority approval. We had 102. The reason we had 102 is we could not find what we thought were good quality estimates for all drugs. So the 102 represents about 40% of all drugs that came through FDA since 1999. And we stratified by first row, fast track versus non-fast track, and looked at the quality gains of the fast-tracked drugs. What you see, and you see it also in the second and third rows for accelerated approval and priority reviews, at least by this metric, FDA seems to be doing pretty well, you could argue. In other words, it's discriminating products that seem to have larger population gains, according to these quality estimates, versus products that go through regular review. And again, this is it's a rather a bit of a complicated story and analysis, and begs probably a full scientific abstract and discussion. But and it's not yet published. But it's another way of trying to put some numbers around policies that we are implementing. And so we're excited about it, and we're going to push it, and eventually we'll publish it. So finally, let me turn to another stream of research that we call predicting coverage. So we've written a number of papers. There's sort of a perspective paper we did, my colleague James Chambers and I, who also was behind some of the other work. Medicare, and for that matter, all payers, have this ongoing challenge, right? New technologies come along. They must make a decision. And for Medicare, and I guess to some extent every payer has a version of this, it's a determination of whether the new innovation is reasonable and necessary. That's actually statutory language for Medicare, as most of you probably know. So we have put into a database all Medicare national coverage decisions. These are exceptions. Medicare makes most decisions at the local level, or Part D plans make their own determinations subject to federal rules. But sometimes Medicare makes these national coverage decisions. Lung cancer screening, colorectal cancer screening, certain Part B drugs go through this process. So we call this playing Moneyball for coverage and reimbursement. In a sense, Moneyball, trying to quantify in baseball and other sports now, with all kinds of statistics, how decisions reflect all kinds of variables. So we've been developing statistical models to try to understand, has Medicare been consistent? The answer seems to be yes, more or less. Is the evidence bar getting higher over time? We had a paper last month in Health Affairs which really strongly suggests that the answer is yes, that technologies which might have been covered 10, 15 years ago with the same evidence would not be covered now. And it's not necessarily that CMS is sort of, tightening up because of fiscal concerns, although probably they are, it may well be that Medicare is just becoming a much more sophisticated consumer of evidence. They're scrutinizing the evidence more tightly. There are other analyses that come out of here as well, and I could share some of that with you. But again, the common theme is we can talk, and we should talk about policies for innovation and measuring value, but through efforts like the two previous speakers mentioned, and I hope these efforts, we can bring some empirical basis to it. So I'll just end on, we should focus on value, of course, not just costs, and that is sort of in a way, a kind of throwaway line, but I think it means really measurement, and we can do it. We can quantify innovation and value with a lot of limitations, but we can do it. And the answers, will depend entirely on the specifics. You can't say new technology is good value or not. You really have to do the studies and make the case and show the data. The information, I think, can inform decisions. But as lots of other speakers earlier today and Mark emphasized, it needs to be combined with changing incentives throughout the healthcare system. So thank you very much. Look forward to your questions.
Thank you very much for having me here. In today's presentations and in some of the discussion earlier, I heard the benefit-risk assessment mentioned several times, and also I've heard references to the therapeutic context, which is what I was going to talk about, even though people really didn't call it by its name. In my talk, what I will do is, I need to advance the slides, sorry. In my talk, I will try to explain the concept of therapeutic context, show how it relates to trial design and timing, and by extension to the cost of drug development. But first, a necessary disclaimer that the views expressed in this presentation are mine and do not necessarily represent an official FDA position. I should also mention that my talk is really about science, and I am an economist. So I will try to take a bird's eye view on some of these issues, and with that, I might miss some of the nuance that's involved in the regulatory decisions and just evaluation of science. So first, I think it's useful to sort of ask, how much does it cost to develop a drug? And you have heard one number, the $2.6 billion. But really, the reality is that it actually varies greatly. So this is from a very recent study by Deloitte. And they studied 12 biopharmaceutical firms in 3 different year cohorts. And what you see in this picture is that there's tremendous difference in the cost to develop an asset from discovery to launch across these 12 companies. And what is captured here is both the direct cost of developing that asset as well as the cost of the failures. And what you can see, the range is from less than 400 million for firm B to firm D, and I don't think you can really, you can see here, firm D over $3 billion to bring one asset on average to market. So what's going on here? So one thing that's going on, and this is actually that article, that report actually gets into, is that some of it is driven by differential R&D productivity. There's growing evidence that small biopharmaceutical firms are more productive than large biopharmaceutical firms, meaning that they get more successes for the amount of money that they spend. What I would like to focus on is to what extent, to really talk about the variance that is related to the kinds of drugs that these companies are developing and the therapeutic context that surrounds that. So what do I mean by therapeutic context? What I mean by this is what the disease is, why it is the way it is, and how we currently treat it. And these 3 are really highly interdependent, but it is really useful to look at them one at a time. So why is therapeutic context relevant? It's because it is really the context for the regulator's determination whether the drug's benefits outweigh the risks. And therapeutic context has implications for R&D because it influences study design. It will determine the endpoints, it will determine the choice of comparator, the entry criteria, the trial size, and the trial duration. It may also influence the timing of trials. So it can help answer questions like, can phase 1 studies, can they be combined with phase 2? If early development establishes efficacy, can an NDA be submitted without completed phase 3 trials? So let's look at the what. What I mean by this is the characteristics of the disease. So how the disease presents itself can really influence the trial design. So for example, chronic and episodic conditions require, in general, they require long studies if no surrogate endpoints are available. It just takes longer to wait for the outcome to happen. Also, for example, does the drug try to prevent an infrequent event? If so, you will need a large sample size simply to gather enough adverse outcomes to be able to compare it across the different study arms. How the disease presents itself can also influence the timing of trials. So for example, phase 1 might be combined with phase 2 if a drug is expected to have toxicity that's unacceptable for healthy volunteers. And this is actually something that happens often in oncology drug development. In some circumstances, the regulator may be willing to approve a drug without completed phase 3 trials if the drug establishes efficacy in earlier trials and the drug treats a severe condition with with few or no treatment options. So the next step is the scientific knowledge, the why of the therapeutic context. Why is the disease the way it is? What is the mechanism of action for the drug? What is the causal pathway of the disease process? So understanding the disease pathophysiology, biochemical and genetic underpinnings of disease helps, number one, directly lower cash costs if companies do not have to do such research in-house. It can lower failure cost by pointing out dead ends. And as Bill Chin actually mentioned earlier, quoting here, we have a lot of failure costs because we don't really know everything. So this is where knowledge actually can really increase the success rates. And this is why having such information in the public domain is really important. The why can help us identify which people are likely to respond or not to respond to the drug, or which ones are likely to experience side effects. So in terms of trial design, knowing who will positively respond often comes with a higher magnitude of an effect, which means you might not need as large of an N of a sample size. It can also cut trial length by helping to establish the causal pathway of the disease process and therefore identify surrogate biomarkers. So Alzheimer's, I think, is a very good example of that last point. So the what of that disease makes it very difficult to study, to develop drugs around. It's a disease, if you lose brain cells, there's really not much you can do. So the focus shifts, understandably, towards prevention and maybe slowing down the disease. But the disease progression takes many years. So again, the what makes it, even on that end, really hard to study. And therefore, you would have to have really long trials unless you have surrogate biomarkers. Unfortunately, we don't really have a full understanding of the disease pathway. Therefore, no appropriate surrogate endpoints have been identified for that disease. On the other hand, you actually have some great success stories. So that really paved the way to many important therapeutic innovation. Just consider the payoff that we have received on building the scientific infrastructure in HIV and cancer. And as a matter of fact, there are spillovers to other therapeutic areas from the infrastructure that was built. So for example, the recent advancements in hepatitis are very much benefiting from all of that infrastructure that was built in HIV. So what I wanted to do at this point is to, no, I skipped a slide somehow. Hopefully I didn't skip it earlier. All right. So the very last component is the how of the therapeutic context, and what I mean by this is how the disease is treated or the existing therapeutic options. Therapeutic options determine the extent of unmet medical need for a given indication. It is, and that's exactly where it becomes relevant in a benefit-risk context. So think about AIDS 20 years ago and now. The extent of unmet medical need has really shifted. So this is actually interesting. This is the aspect of therapeutic context where we can become victims of our own success. Some conditions have established standards of care with existing therapeutic alternatives, and in those cases, trials could not be done relative to placebo because withdrawing effective treatment from patients would actually be unethical. So if a standard of care is effective, they can really set a high bar for drugs that would try to compete with them. And so in that case, the need to establish superiority or non-inferiority will drive the sample size up. So what I would like to do at this point is help you visualize the therapeutic context in the clinical development process. So what I'm going to show you is 3 examples of drugs in clinicaltrials.gov, and this is basically a mock-up to help you understand what I will be showing you. So what you will see is that the horizontal bars are going to be the different trials that are reported in clinicaltrials.gov, and keep in mind is that not every trial might be reported, and they are the colors represent the different phases. The gold circles are the pivotal trials. So those are the ones that were being used to support the submission, the NDA submission. The black line, the vertical black line is the NDA approval date, and you can actually see the timing from relative to that. And the dashed line, the dashed line is the NDA submission. All right. And one thing to mention is in all the 3 slides that I'm going to show you next, the scale is going to be the same. So whatever differences you observe in terms of the size of the trial or the length of the trial is all on the same scale. So first, let me talk about Brilinta. So Brilinta, what you see here on the left-hand side is a study with over 18,000 patients. Brilinta was trying to establish, they wanted to establish that they can reduce the rate of specific cardiovascular events in patients with acute coronary syndrome or ACS. So the sponsor used a pretty standard endpoint for cardiovascular studies. It's a combined endpoint of cardiovascular death, stroke, and heart attacks. So all the studied patients have ACS, but we expect maybe a fraction of them to actually have one of these events, maybe 10%. So to deal with this, you really have to increase the size so that you can get enough of the adverse events to compare it to the control arm of the study. Another reason that's driving the large study size is that the sponsor is trying to establish superiority relative to Plavix. They're looking for a possibly really small improvement in efficacy, maybe a couple of percentage points at best. To find that small of an effect, they have to power the study and they have to really increase the number of patients by even more so. Then the drug gets approved, and then they look for another indication. Same endpoint, but a different patient population. Because the baseline rate of adverse events in that population is lower than in ACS, you have an even larger sample size. So now let me show you something on the very opposite end of the spectrum. This is Blincyto. It is a poster child for an accelerated approval drug. This drug was seeking an indication to treat relapsed patients with acute lymphoblastic leukemia or ALL, which is a fatal disorder. Historically, the response rate for these patients to single drug therapies is really poor, about 5 to 12%. Using combination therapy with sometimes up to 5 chemotherapy treatments and a stem cell transplant, the remission rates improved to maybe 25-46%, but even their median survival rate is about 3 to 6 months. So given the really dismal prospects that these patients are facing, it really did make sense to do an open label single-arm trial, which is really a trial where all the patients get the drug. The complete response rate for this trial was 32%, which is significantly better than any other single drug. The outcomes were about the same as with combination therapies, but taking one drug was sure better than having 4 to 5 chemotherapy drugs and maybe a stem cell transplant. So given this therapeutic context, the benefits of the drug appeared to outweigh the risks with that study alone. And so the drug got accelerated approval without phase 3 studies completed. Those phase studies continued on, and they're used to be used as confirmatory studies for full approval. The last example I want to show you is Sovaldi. The picture here looks a bit different. So one is because hepatitis C virus is now very well understood, drugs can be designed to target specific steps in the virus life cycle. And so presumably you would expect potentially a higher magnitude of an effect if you really understand the disease. So what's striking is the number of studies that they conduct here. The reason is that they designed studies to show efficacy across several different genotypes of hepatitis C, and they also were stratifying on the patient's prior experience with interferon-based therapy. Also, they needed several studies to establish the appropriate duration of treatment. They could have done this probably with one, but the sponsor chose several. So the very last thing I wanted to show you is a summary of just one metric related to drug development, and that is enrollment in pivotal trials. So again, the pivotal trials are the adequate, well-controlled studies, and this is the total patient enrollment in the studies that were supporting the different submissions. As you can see, and perhaps this is not a surprise, you see a large variation across these different therapeutic areas. But I think what you might be more surprised by is to see the tremendous variation within some of these areas. But what's going on here is the therapeutic context. So these groups, these drugs are not homogeneous when it comes to the therapeutic context. So some of these drugs in a given group are trying to prevent a low probability outcome, and some don't. Some of them, we have an understanding of the disease and we can identify responders, and in some we don't. With some of these drugs in the class, and I don't know how this possibly happened. All right. With some of these drugs in the class, we have good treatment options, and with some, we don't. So I guess this is what I wanted to offer as a thought, is that the implication for researchers is if you're trying to understand the cost of drug development, the drug development timelines, you really have to account for the therapeutic context, for the what, why, and how is it treated currently. And if you were to ask me what are the implications for policy, I would say the what is given, we can't control it. The how we treat it is given at right now. The only element out of these three is the why, and that's something sort of the only area where we as a society have control. So I wanted to end with that. Thank you.
I'd like to thank all of the presenters and invite them up to the panel. I know we are running over time, but we will spend about 10 or 15 minutes for the panel discussion. And while you're getting mic'd, I'll at least try to jump into some of the questions. So listen as you're getting mic'd. We did hear a lot of interesting results. Murray started off with some not necessarily depressing results, but he did paint a picture of the fact that we are seeing declining lifetime sales, potentially negative economic returns after tax, decreasing in preclinical activity. So this sort of says to me that there's a problem. But then we heard from the other panelists that well, it's not that straightforward. It's very complex to really understand necessarily, is that a bad thing and what's really driving that. So what I would like to ask all of the panelists is one question, and then I will turn it over to the audience. But quite frankly, what does the data tell us, and what should we be measuring? So this morning we heard on the first panel a lot of ideas on improving clinical trial efficiency, about the potential roles of real-world evidence development, and the importance of bringing in patient perspectives. Is that anecdotal? Are improvements in those areas anecdotal? You all are experts in the data and can help us understand, are those the right focus areas based on your experience with your data and looking at what's really driving innovation? What we talked about this morning and a lot of the proposals that you might have seen in the 21st Century Cures, are those the right area based on what you're seeing in the data? So Murray, do you want to?
Well, I would say they are good areas to focus on. What we need to know is, what we need to be measuring, though, is what impact that they are having, and put that in the context of, I think, what Marta took us through, which is the nature of the area of research, because clearly things differ dramatically depending on where that focus of innovation is placed. So I think this speaks to the need for us to be able to systematically look at these areas, look at their implementation, and be able to measure what's actually happening in terms of the level of improvement relative to the innovation in different therapeutic classes.
Yeah, so I would say that when you listen to the public policy discussion and the debate around what to do to improve biomedical innovation, the answers fall into certain consistent categories, and they're the same ones year after year after year. The details of proposed solutions may differ, but they're the same basic points that people are trying to influence in the system. I mean, people talk about proposals to reduce the time that it takes to develop a drug or reduce the cost, and people talked a lot about that on the first panel with respect to clinical trials and the inefficiencies that maybe we can design out of our clinical trials. But time and cost are always very, very high on the list. Incentives, so people always talk about patents and exclusivity, regulatory exclusivity, and how do we create better incentives for these investments to be made. And then, of course, money for research. More dollars are always better. And so the solutions fall into a consistent set of buckets, and there's no doubt that those are the right buckets. If you want to drive more innovation, you want to make it happen faster, you want to make it cost less, you want to make it more valuable to the innovator to innovate, and that'll drive more investment in innovation and more output through the system. But the real challenge is differentiating among the different proposals that come about to try and move each of these things. And probably just about all the proposals that are out there will have some positive impact on one or more parameter in the system. All of them also have some costs. They cost money to the system. Maybe they're imposing more burden on FDA, and that has costs or will spill over to other aspects of what FDA does. Or obviously extra exclusivity costs the system money in paying for drugs. So figuring out which ones to prioritize, which ones will actually work, which ones will have a benefit that will exceed the costs associated with putting them into place is where we start to struggle. And that sort of leads me back to the whole discussion we're having here on this panel is how do we get our arms around measuring and assessing the impact of each of these kinds of policy interventions or figuring out are we intervening at the most impactful spot in the system. So that's really where I hope we can go and sort of synthesize a lot of the kinds of different inputs that we all talked about here and put it together in a way that allows the community to ask questions that we're not even thinking of up here, but shed light on all these policy issues.
Marta? Or?
So what I would say is sort of going back to the concept of streamlining clinical trials. When you look at clinicaltrials.gov, what you see is when the trial started and when it ended. And it's really hard to tease out how much of the length of the trial is driven by the therapeutic context and how much of it is driven by inefficiency in the system. So for example, it is true that with cardiovascular drugs, it's not as hard to recruit patients into these. Probably with Soval, they didn't have many problems. But with the cancer drug that I mentioned, they probably had a lot of trouble doing this. It actually would be really interesting to go back and try to adjust for the duration of treatment, sort of take the clinical trials that were designed and then also actually look how long a given patient had to be treated, and then to see how much of the difference is, and that would capture the inefficiency in the system in a sense. So that I don't know that has been done, but I think it's doable.
Here?
I guess I would just add that there are a series of policies that we might put under, to oversimplify, informational policies. Better information for the decision makers, giving them information on value, public and private, from cost effective. Mini Sentinel, you might argue, is informational. I think clinicaltrials.gov possibly put under there. Covered with evidence development, collecting additional information. That is information without necessarily changing regulatory processes or incentives, which is the second maybe big category, which is harder and probably more impactful. And I think we need both. But at least I hope from this last session, there's a lot more we could do on the information side, measurement, tracking, providing that information to inform policies.
Yeah, a lot of the proposals are in fact intended to improve efficiency of clinical development to get promising therapies to patients sooner. But implicit in all of that is that we are developing therapies that are impactful to patients and that mean something to patients. And we do a very bad job, I think it's been clear, of really measuring that. And cost per qualities and increases in qualities is an important measure, but it's hard to do. So my question, just stemming off of that, are how hard is it, and are there potential other ways or different ways of measuring qualities that can be more widespread, more incorporated into the learning of patient experiences as we go along once a product is approved?
Right. Well, certainly there are methodological challenges in measuring qualities and cost per qualities. I don't think they're insurmountable, or compared to many other things we measure, terribly sort of burdensome. There certainly are other metrics we would like to have alongside those. There are lots of ways to measure health. Instead of qualities, we can measure life years. We can measure with disease-specific metrics. We could measure, costs and consequences and leave it as sort of disaggregated information, not put it into a ratio. There might be lots of other information we'd like to add to the cost per quality ratios. Cost per qualities, as I think everybody knows, has been controversial. Raises the specter of rationing. It involves some strong assumptions and so forth. But implicitly, we're making decisions, and we are spending our money, and allocating our resources given constraints. So we either use information like cost per quality and other metrics, or we don't, but we're still making these decisions. So I'm a great advocate for information like this, but by no means is it the only metric to use. I think there are others.
Any other comments on that point? No.
I would just bring it back to the patient role in contributing to the definition of quality or whatever the particular metric is, and I recognize it's at least implicitly already part of the quality measure. But I think as we hear more discussion about the patient role in reporting outcomes and defining outcomes and so on, it may be a good opportunity to also reframe part of the quality metric to reflect that patient input.
So with this, I'd like to turn it over to the audience. A question in the back.
My question's for Marta. Thank you for a very interesting presentation. But I want to make sure I understand one of the points that you made. It sounded like as treatment gets better, if you want to then have an additional treatment, you're going to need a larger clinical trial. And if that's the case, then you would drive up, as you get better and better treatments, the cost of an additional treatment will go up, right? So eventually that will, drive the cost of treatment up, and also the return on investment will go down, so ultimately it would sort of cut research off. Was that the implications of what you were trying to say?
No, not necessarily. I mean, if the alternative is really good, and that's the baseline from which you're going, if you're comparing it to a sugar pill versus if you're comparing it to an effective therapy, then you're looking for a small effect. If you're looking for a really large effect, you don't need a large sample size. If you come in, there are existing therapeutic options for hepatitis, but they were probably expecting a really large magnitude of an effect for the reasons because they understand the science and they understand how the drug works and so on. So that's not necessarily one direction in how that goes. But if the treatments are effective and you're just trying to squeak out from it's purely statistics. It's if you want to show statistically that you are having a statistically significant difference, and the difference is going to be tiny, you will have to have a large sample size to do that. It's sort of reality of statistics.
I think that's all we have time for questions, so I'm going to go ahead and wrap up this panel. Thank you to all of the panelists for your great comments. It's excellent data. And I'll turn it back over to Mark for just some final comments. Thanks.
Thank you all very much. I'm going to be very brief, so you can just sit tight for a second. We've covered a lot of ground today. There are obviously a lot of unresolved issues and challenges in biomedical innovations, but a huge amount of interest and a huge amount of activity in trying to address those challenges. I was particularly impressed with the work of the last panel in describing how to put some measures of progress on the efficiency of the biomedical enterprise and the productivity of the biomedical enterprise through the development stages, the cost, the revenues, the value of treatment, and how all of these are influenced by therapeutic context. So this, in turn, provides a way to help guide further policy development. But as you heard from the last panel, there is still more work to do to connect the two. I'm very pleased to see the progress, though. So we've covered a lot of ground today, and I just want to end by thanking all of our panelists in this session and the first session for framing the big issues in biomedical innovation. We are obviously, like many of you in this room and joining us online, we're going to continue to be working hard on these very important issues for protecting and promoting the health of the public. I'd also like to thank the staff at Brookings who helped put together this event. You all have already heard from Greg Daniel, but also Serena Coates, Matt Longo, Christina Flores, Joanna Klassman, especially Morgan Romine for an extraordinary amount of work to get this event together. We look forward to seeing all of you again soon and look forward to more progress in improving biomedical innovation in the United States and around the world. Thank you all very much.